Vanakkam friends, Dr. Vanangamudi on behalf of Trinity Cultural Academy Coimbatore and New India Publishing Agency New Delhi welcomes all of you for this presentation. And this presentation is on self-incompatibility, and this is part 1. It comes under the main subject of plant breeding and genetics. This is 11th series of this presentation, and we will be seeing the topic of interest today. What do you mean by self-incompatibility? It refers to the inability of fertile pollen to fertilize its own egg that will result in failure of seed set. And self-incompatibility occurs at any stage between pollination and fertilization. It is reported in more than 6,000 species in the members of Leguminosae, Rosaceae, Solanaceae, Compositae, Brassicaceae and Graminae. And as far as the distribution of self-incompatibility is concerned, there are about 6,000 species. This mechanism exists, and it consists of 250 genera, 70 families, and 19 orders, both in monocots and dicots. For example, in plants with self-incompatibility, when the pollen grain reaches the stigma of the same plant or another plant with a similar genotype, this you have to note down. The pollen germination, pollen tube growth, ovule fertilization and embryo development are halted at any stage between the pollination and fertilization, and consequently no seeds are produced. And this picture clearly indicates that this is having the same genotype, and if the pollen of this same genotype falls on the stigma of the same flower or the plant, that there is a self-incompatibility mechanism exists. If the same genotype SX, if the pollen grains will fall on the SY stigma, that is a different genotype, and there is a compatibility and pollen tube growth takes place. And regarding the interspecific incompatibility, if the pollen grain from Brassica oleracea and falls or pollinated on the stigma of Brassica rapa, and there is the interspecific incompatibility and there is no pollen germination is noticed. Regarding the general features of cell compatibility, it prevents selfing and it promotes only the cross pollination, that is otherwise called as outbreeding, that increases the probability of new gene combination. It may cause any changes in morphological, physiological, genetical and biochemical characters, and normal seed set is happening only on cross pollination or outbreeding. It may operate in different stages, as I told, right from pollination to fertilization. It reduces the homozygosity, and that means it increases the heterozygosity. In plants, cell compatibility is inherited by a single gene with different alleles, namely S1, S2 and S3. And self-incompatibility mechanism was confirmed in different crops like kale by Thomson 1957, in radish by Sampson 1957, broccoli by Sampson 1957 and Odland 1962. In cabbage, Adamson 1965 reported self-compatibility, whereas Hosen and Croix 1979 reported self-incompatibility in cauliflower. And as far as classification of self-compatibility is concerned, based upon the flower morphology, that is heteromorphic self-compatibility, and there are two types in this category, that is distyly and tristyly. And regarding homomorphic self-incompatibility, there is again two divisions: sporophytic incompatibility and the gametophytic self-incompatibility, and that will be seeing in the next few slides. Regarding the genes involved, based on that, it is monoallelic, that is governed by single gene, and the second group is diallelics, that is governed by two genes, and polyallelics governed by many alleles. As far as the cytology of the pollen is concerned, and there are two types, that is binucleate, there are two nuclei are involved in the pollen, and trinucleate means pollens with three nuclei. Regarding the site of expression, and there is a stigmatic a self-incompatibility, that means expression site is stigma, and style or incompatibility, that is expression site is style. And if the expression site is ovary, that is grouped under ovarian group. And types of self-compatibility, there are 5 types are there. The first one is single locus self-incompatibility, and under this there are two types: gametophytic self-incompatibility and sporophytic self-incompatibility. And these two types only we will be seeing in this presentation. And the type two locus gametophytic self-compatibility, and third heteromorphic self-incompatibility, and fourth one cryptic self-incompatibility, and fifth one late acting self-incompatibility. These four we will be seeing in the part 2, that is the next presentation. And look into the single locus self-incompatibility, and as I told, there are two types, that is homomorphic system, and this is governed by only one type of flower. It is compatible, and incompatible plants cannot be differentiated on the basis of floral morphology. There are two types of incompatibility in single locus incompatibility. First one is gametophytic incompatibility, and next one is sporophytic incompatibility. First we will see the gametophytic incompatibility. This was first reported by East and Mangeldorf 1925 in Nicotiana Sandere, and it is governed by multiple alleles of S gene pollen. About 21 multiple alleles have been found to control incompatibility mechanism in gametophytic self-incompatibility. These alleles are independent in that action and do not show any dominant relationship. The pollen and ovule carrying dissimilar S alleles can set if the style does not inhibit the tube growth, that means pollen tube growth. The tissue is diploid in nature, and if there is any S alleles are common in pollen, it arrests the tube growth. Regarding gametophytic self-incompatibility, and the S alleles are produced or synthesized only after completion of meiosis. This is more important, and this is the pollen cell from which the pollen grains are formed. There are two types of S1 and S2 product. This is the S1 product, and this one is the S2 product of mature pollen, that is bicellular. The example, the styles of S1 genotype. The styles of S1 genotype would not allow pollen tube of S1 or other alleles to penetrate. That means it does not germinate, whereas it will not inhibit the S3 pollen tubes. It will not inhibit the S3 pollen tubes that are more important. And there are three situations here. Fully incompatible. This is fully incompatible. Here the S1 and S2 pollen cannot germinate in S1, S2 stigma or style, and this is partially incompatible. And here the S1, S2 pollen will partially germinate in S1 and S3 style or the stigma, and fully incompatible means the S1 and S2 both the pollen will put forth the pollen tubes in S3 S4 genotype, that is the style and the stigma. And the examples for this self-compatibility, the one half of the families of angiosperm, like the Solanaceae family, potato and tomatoes, and tomato both wild and not cultivated cultivars are also having self-incompatibility mechanism. And the tobacco is another example. The petunias, beet, that's a beta vulgaris, buttercups, ranunculus, lilies, roses, many grasses, pear, peach, and lucerne. And in these crops, the gametophytic self-incompatibility mechanism exists. And next we will be seeing the sporophytic incompatibility. It was first described by Hoggs and Popcock in 1950 by in-crop Crepus and by Gestel 1950 in Parthenium. This system is also governed by multiple alleles of S gene. Tube reaction is governed by the genotype where they are produced. For example, both type of pollen produced by S1 and S2 plant would have similar genotype, and inhibition occurs at the stigmatic surface, and the pollen does not germinate, and it will not put forth the pollen tube in the on stigma. In this system, rejection of self pollen is controlled by the diploid genotypes of the sporophyte generation. The main difference between the sporophytic germination and gametophytic germination, here the S allele products are synthesized before the completion of meiosis. This will happen only in sporophytic, whereas in gametophytic, this S allele products are synthesized after the completion of meiosis. This is the major difference between the gametophytic and sporophytic incompatibility. Here, the S1, S2 pollen will not put forth the pollen tube that is self-incompatible with the S1, S2 type of genotype. And here, the S1, S2 will also have incompatibility mechanism yes in S1, S3 in this mechanism, whereas in gametophytic this will have partial compatibility. This is another major difference between the gametophytic and sporophytic. Sporophytic, the S1, S2 pollen will not germinate, will not put forth the pollen tube even at S1, S3, and S1, S2 is fully compatible with S3 and S4. The examples for this type of incompatibility is members of mustard family, that is brassica, including turnips, rap, cabbage, cauliflower, and broccoli. I'm sorry, not cabbage, that is cauliflower, cabbage, and broccoli. And there are different mechanisms of self-incompatibility are involved. There are three categories. First one is the pollen stigma interaction, and second mechanism is pollen tube style interaction, and third mechanism is pollen tube and ovule interaction. We will see one by one. The pollen stigma interaction, it occurs just after the pollen grains reaches the stigma, and generally it prevents pollen from germination. There is no pollen germination at all. Previously, it was thought that binucleate condition of pollen in gametophytic self-incompatibility and trinucleate condition in sporophytic self-incompatibility was the reason for self-incompatibility, but later on it was observed that they are not the reason for self-incompatibility. Under homomorphic incompatibility, there are differences in the stigmatic surface which prevents the pollen to germinate. And in gametophytic system, the stigma surface is plumose, having elongated receptive cells, which is commonly known as wet stigma. The pollen grain germinates on reaching the stigma, and incompatibility reaction occurs at the latter stages only. That means when the pollen germinate and the pollen growth enters in the style and also in the ovule, and there the incompatibility mechanism takes place. Within few minutes of reaching stigmatic surface, the pollen releases an exins exudate, and this will either having protein or glyceroprotein. This reacts with pellicel that induces callus formation, which further prevents the growth of the pollen tube. Before that, in the sporophytic system, the stigma is papillate, whereas in gametophytic system, the stigma is wet. In the sporophytic system, the stigma is papillate and dry, and it is covered with hydrated layer of protein known as pellicel, which is involved in incompatibility mechanism. As soon as the pollens are reaching the stigmatic surface, the pollen releases exins exudate, which is either protein or glyceroprotein. This will react with pellicel and induces callus formation, which further prevents the growth of the pollen tube. The second mechanism is pollen tube style interaction. Pollen grain germinate and pollen tube generally penetrates the stigmatic surface, but the growth of the pollen tube is retarded with the style as in petunia, lycopersicum and lilium. These are all the example. Here the protein polysaccharide synthesis in the pollen tube stops, resulting in bursting of the pollen tube, that leading to the death of the pollen nuclei. So there is no fertilization at all. The third mechanism is pollen tube ovule interaction. In Theobroma coco, pollen tube reaches the ovule and fertilization occurs, but the embryo degenerates later due to some biochemical reaction. So the embryo degenerates after fertilization. And the last aspect is pseudo incompatibility. It refers that incompatibility plant shows some degree of self fertilization. Thus, so it is used for creating homozygous S lines and maintain them by selfing. Thank you very much for listening this presentation. Kindly watch these videos, like, share and give your valuable comments and subscribe the YouTube channel. Thank you very much.